Great. So good afternoon, everyone. I'm happy to be here to share my solution to this challenge, and I'm also happy to know that being able to come really helped. So and also one disclaimer: if the previous team's solution using that binarization is illegal, then mine is illegal too. So me personally, I'm super interested in single-cell analysis, and when I first learned about this challenge, I was like, okay, I have to jump in. And well, let's jump into my solution. So what I did was I call it similarity-preserving based gene set selection for doing this kind of mapping. And before going to my solution, I'd like to kind of quickly walk through the data so that I can also share with you guys my thought process when I was thinking about this challenge. So the amount of data that are provided are, in my mind, everything is like matrices and boxes. So it's one piece of the data is the single-cell RNA-seq data of roughly 1,300 genes across 1,300 cells across many genes, and there's the raw counts version of the data, the normalized version of the data, and the binarized version of the data. And then also the in situ data for 3,000-ish locations across 84 genes, and again, raw data and binarized data, and also the XYZ coordinates. And the ground truth is basically looking at the binarized version of the RNA-seq data and the binarized version of the in situ data of the 84 genes altogether to build a similarity mapping and hence map locations. So now, reading the challenge, this is about predicting the locations of these cells using fewer genes. But thinking about it a little bit more, actually, the question is, how can we do the prediction using a subset of genes that best approximate the prediction done based on the 84 genes, because the 84 genes is the ground truth. And thinking about that even a little bit more, especially after the webinar and some initial discussions of what data can be used and what data cannot be used. So this turned out to be, well, in my mind, a feature selection problem. How can we select meaningful, spatially related subsets of genes without knowing any in situ data at the beginning? So that's basically saying that, when we're trying to select genes, the only available information to us is the single-cell RNA-seq data, and plus the knowledge of the list of the 84 genes. Okay? And we have to, at that stage, pretend that the in situ data haven't been collected yet. And then later in the sort of the location mapping stage, we will only use the in situ data of the selected subset of genes, as if these selected subset of genes, their in situ data is collected after we've finished feature selection. And therefore, for the in situ data corresponding to the genes that were not selected, we need to pretend that they don't exist at all, because they were never ordered, they were never collected. Okay, so now the basic idea of sort of my feature selection strategy actually resonates a lot with the previous talk related to continuity. And so thinking about this, thinking about the ground truth, what we do know is that for two cells that are very similar to each other according to the 84 genes single-cell RNA-seq data. Okay, so in the single-cell RNA-seq space, according to the 84 genes, for two cells that are very similar to each other, they will be mapped to similar locations. For two cells that are different, they may be mapped to nearby locations. They may be mapped to very different locations. We don't know. But for the thing we do know is, again, for two cells that are similar in the single-cell RNA-seq space according to the 84 genes, they are going to be mapped to a similar location, okay? And now for our feature selection, well, what we would like to do is that, I think, or what I would like to do, is to find an optimal subset of these 84 genes that optimally preserves such relationship, so that basically for two cells that are very similar to each other in the single-cell RNA-seq space according to the 84 genes, I would like them to be also very similar to each other in the single-cell RNA-seq space according to a subset of selected genes. Okay, so I want to select subsets of genes that best preserve the sort of the continuity or the similarity between very similar pairs of cells. All right, so here's kind of a walkthrough of my solution, and it's on Synapse, so I'll try to go through it quickly. So the first step is kind of not a gene selection, but a kind of a gene maybe enrichment, because single-cell RNA-seq data is noisy, there's dropouts. So similarities based on the 84 genes may not be best. Is there a way for us to even enrich that set and work with more genes to define cell-cell similarity? So what I did was that for every gene I tried to ask, can this additional gene be well predicted by the 84 genes using the single-cell RNA-seq data? If yes, then there might be some additional genes that are also spatially related, similar to the 84. Okay, so I did basically some linear regression analysis, some randomization, some thresholding, and at the end of this step, I arrived at a list of 321 genes, which include the 84. And these 321 genes, they are all, I think, are spatially related, spatially relevant genes. I call them landmark relevant genes. And this step of analysis is done on the normalized version of the single-cell RNA-seq data only. So after that, I'm working with 321 genes only. And again, I'm looking at the normalized single-cell RNA-seq data at the second step. And what I did was that I built a nearest neighbor graph, a cell-cell graph that encodes similarity, encodes the continuity. In this step, there was involved is some normalization, some PCA, and some k-nearest neighbor graph. And the output or the result of this step is a cell-to-cell graph where connected neighbors should be very similar according to the 321 genes and hence very similar according to the 84 genes. Again, in the single-cell RNA-seq space. And after that, what I needed is I want to do feature selection, so I need to have a score to kind of score gene subsets. So what I did was that, if we work with the binarized version of the single-cell RNA-seq data, because the final mapping is on the binary data. So what I did was that for every cell, so if we focus on just a particular subset of genes, whatever subset, 20 genes, 80 genes, 100 genes, doesn't matter. For a particular subset of genes, I can use the binarized version of the single-cell RNA-seq data and compute a cell-to-cell difference or cell-to-cell similarity. And then I derived a score here. Basically, for every cell, for one cell, I will rank order the similarity of all the other cells to this cell. And then I will look at this cell's direct neighbor in the graph that I just built and see for these neighbors, do they have top rank? Does the sum of their rank give me a very small value? If yes, then the similarity encoded by this particular gene set that I'm testing agrees with the similarity graph that I built previously, which I would like to preserve. Okay? So with this scoring, this is like my big gun to solve the challenge. I could use that to score gene subsets. So I start with the 84 genes, okay? And again, this is using single-cell RNA-seq data only, the binarized version. So I begin with the 84 genes, and then I try to remove each of them. So there are 83 gene sets. So for each 83 gene sets, I would use that score to see how well a 83 gene set preserves the distance metric or the graph that I just built. So looking at those 84 possibilities, I select the best one, and hence one of the 84 is gone. And the one that's removed is the least relevant gene that I would like to work with. So now I have 83 genes, and then I can try to remove one of them and look at 82 gene sets, and then see which one is the best so that I can kick out another one. And the second one is basically the second least important gene. And I can keep doing that, kicking out genes from gene sets, keep reducing the size of the gene set that I'm working with, and that would induce a ranking list of all the 84 genes. I actually do it all the way to the, only one gene is left. And this ranking list is the basis of my submission for all the 3 challenges. So once I have the ranking list, then I can take, for example, the top 20 or top 40, doesn't matter. So I can take, for example, the top 20 gene, and then I will take the binarized single-cell RNA-seq data of the 20 genes only, and the binarized the binarized in situ data for, again, the 20 genes only. And I have these 2 binarized versions. I used F measure instead of MCC. I forgot why I chose it. maybe I didn't read the challenge correct, and I thought the ground truth was based on F measure, but it doesn't really matter. So I used F measure to compute a 1300 by 3000 kind of similarity matrix, which served the mapping, served as the basis of the mapping, and hence identify the 10 most probable locations. And those were the 10 locations that the submission reported. Okay, so quick discussion. So the reason why I think my solution is a little bit interesting is that this is a unified solution for all the three sub-challenges. So I actually did gene selection only once, or gene ranking only once. And I feel that building off of, say, this solution or the challenge, potential ways to kind of improve the biology is that maybe you can try to develop computational algorithms to define the initial set of 84 genes. Maybe these 84 are not the best 84, but is there a way for us to really work in a scenario where we actually know nothing about what possible in situ is available, even the list of 84? And also, the performance of the final mapping, I did it using the binarized data because the ground truth is based on binarized data, but maybe using continuous data might be better, who knows? And I think a potential way to kind of score better in the challenge might be that, the initial step where I enlarge the set of 84 to 300 something, maybe that's actually a bad choice, because the ground truth and the similarity that the challenge is trying to score is based on the 84. So enlarging it might hurt the performance a little bit in terms of what score we get. But then again, I think in terms of doing the biology right, maybe it's better to enlarge it. And another possibility is, as what was presented just now, a little bit of post-processing of the 10 locations that were identified. I think that would be an idea to kind of, improve the stability and shorten sort of the distance between the 10 submitted locations. I didn't think about that, but I actually think that's a good idea. And that matches well with the scoring function. So that's all I want to share. Thank you very much.
Thank you. Very nice. I have a question. When you did you chose the 20, say, and then you used the 20 in situs to define the mapping better. Did you do that only once in all your attempts, or did you do that more than once? Because if you did it more than once, there is danger that you have leaked data into, right? So it's like once you committed the 20, then you have to use it once and never more. Can you say whether you used it once or more?
Okay, so to be honest, I did it twice. But these two times I used completely two different approaches. So the approach that I'm presenting is what I eventually submitted. But then during the midterm submission, I actually submitted another solution that got slightly a worse score. And that was mainly based on distances or kind of entropy. So, and that's a forward selection process where I first, so based on the binarized data of the 84 gene in situ genes only, but of course based on the binarized data of the single RNA-seq. The first gene I select is the gene that has the highest entropy. So if there's an all zero gene, it's useless to me. Right? So the first gene is the one that has the highest entropy. And the second one is the gene that is most independent from the first gene. So in essence, the second gene is the one that's most distant from the first gene. And the third one is the most distance from these two, et cetera, et cetera. So that forward selection process also induces a ranking, which I can take, say, the top 20 genes to do one submission, top 40, top 60. So that was one algorithm that I did. And then this was the other. I submitted both for the midterm submission, and I didn't really change anything after that.
Okay.
So one thing I find very interesting is that you try to compute genes which are informative about spatial organization. And so, of course, in the Drosophila embryo, we know that this is, at this point in development, it's transcription that is defining patterns. And the 84 genes in the Berkeley database are not chosen randomly. I think they're almost without exception transcription factors, and highly expressed or expressed in the early embryo. So a simple test for your 300 plus that you found, they should be highly enriched in transcription factors. Is that true? And did you find maybe some novel genes that you think are really important and have been overlooked? So?
I haven't tried it, but great idea. This is the first thing that I'm going to look at tonight.
Okay.
Well, this is the kind of things that we should do when we write the paper about the challenge, right? The biology.
Thank you. Hi, I have one question, but I write it here, so I'm gonna sit and read it. Because you said that you have selected 321 genes, right? And did you somehow analyze those genes to determine this rank that you said that you start with 84, then remove 1, then 83, then remove 1? So what I'm trying to say is that maybe you can do that for your 321 genes in order to determine like a bigger rank, and then you can somehow remove these 84 genes that are well known. Well, I don't know if they are well known, but you can remove it or put them aside for a while, and then try to analyze from a biological point of view these other genes in terms of your rank. That could be very interesting.
Yeah, I think that's an excellent suggestion. And if I understand you 100% right, I think what you're suggesting is maybe the selection process does not have to start with the 84. Maybe we should select it with the 321, do the selection, and then we can actually look at whether the 84 survive the process and later eventually end up at the top. And if the answer is yes, that would be a confirmation that these 84 are really well selected. But then if, for example, some other gene outside the list of 84 kind of, eventually end up still on top when I do the selection from the 321, then that would be a suggestion that maybe, there are interesting genes that should have been included in that database. Yeah.
So, so the common feature between the first presentation and the second is that you didn't use in any way the information of the location of the cells. So it was just clustering the genes. And I think that was a great approach. For instance, I was focused on trying to find which of the 84 genes would predict those locations. And initially we were ranked first on the top challenge, but then the discussion started to come in that, well, you should make sure that you don't overfit. So, we said, well, then we probably need to do some cross-validation. And that made us to lose the chance to try anything else, because now doing cross-validation, you are going to be worse at the prediction. And then, but I think the idea of really clustering genes and don't even using anything about the position, that was excellent. I think now in hindsight, I would have said to Pablo to say, don't even say that you can use the location of those genes. Forget about that. But
And the question was, no, that's okay. Right, so, but I think that goes to what Nicolaus was suggesting, that there is a principle of organization that is in the data itself that you can try to extract, and that, but a priori you don't know that, right? So there is a question there, and then I come to you.
So very nice work, and very nice the idea of clustering. So I have one comment and one question. So about overfitting that was mentioned before, I guess the concept of overfitting is more general. It's not only if you use the Y values. So the question is, how do you know that this method can generalize, given the data that we have, the data that you use? This is one question. The answer, and I have also a comment. Yeah.
Okay, so thinking about speaking from kind of a non-rigorous mathematic term, that when we're trying to do something, we always want to do it in a way that's sufficient and necessary, so that would be the best solution. And right now, the solution is based on the continuity, which is actually just a necessary condition, but not sufficient. So I think for a really good solution, okay, in that really good solution, this kind of continuity or neighborhood distance preserving has to be there. But a solution that has these neighborhood preserving property, is it sufficient to lead to an extremely good solution? Actually, the answer is no. So I have to admit that this is sort of, kind of throwing a dart half blindfolded. So I know that I'm using something that I'm using a property that a good solution will have, but then is this property sufficient to lead to the good solution? Not necessary. Not necessarily. So was there a second question? Or we can do it offline.
There is a reception coming up, so let's continue this discussion after in the reception. But now,
I just want to say that I'm not really in the community, but there were really interesting comments. So when the paper is written, hopefully, probably that gets sent around so that people can put in their comments. So that I think would be great.
Yeah. Maybe we can continue this discussion after the next talk, but thank you very much.